Hello everyone, welcome to my YouTube channel. This is Soumya. In this video, we are going to learn a plant hormone that is gibberellins. I'm going to start with a short introduction and then we will move on to the definition and then occurrence, and also I'm going to explain the biosynthesis in this video. What are these gibberellins? Gibberellins are plant promoters. In plant hormones or phytohormones, there are mainly two categories that is plant growth promoters and plant growth inhibitors, and this gibberellin is a plant growth promoter. Gibberellins are represented by GA, okay, capital GA, and it represents a class of diterpenoids, and it is a fungal hormone. Why these are called as fungal hormones? I will explain why these are called as fungal hormones. Just stay in this video till the end. Historically, the discovery of the gibberellins can be traced back to the 1930s in Japan, when scientists were investigated a peculiar rice disease known as a bekane or foolish seedling. See, to explain this second point, the discovery of this gibberellins is very interesting because a Japan farmer was working on his rice field. He found out that some of the rice plants were very taller and they are pale in color and they are seedless. See, he observed why some of them are like this. So then he got to know that these plants are got affected. Okay, they got affected by a disease. Okay, that is a fungus disease. Okay, that is called as foolish seedling. Okay, see, as you can see in the diagram here, you can see this is the normal rice plant. Okay, and you can see here this plant is a pale yellow in color and these seeds are not real seeds. Okay, so that's why it is called as a foolish seedling. Okay, there will be no seeds inside. So this plant is got affected with the fungus. That's why it is named as bekane or foolish seedling. And why the rice has been affected? This is because a causative agent. The causative agent behind this disease was identified as the fungus, and that is called as Gibberella fusicori by a Japanese scientist named Ichai Kurosawa in 1926. A rice ಎಲ್ಲ ಆ ಥರ ಆಗಿರೋದಕ್ಕೆ ಕಾರಣ ಏನಪ್ಪಾ ಅಂದ್ರೆ because of the fungus. That fungus is called as Gibberella fusicori. These gibberellins are synthesized within the plastids of the plant cell, and then organelles that is integral to the food productions. Like if you look at the biosynthesis, gibberellins biosynthesis is going to start with the plastid, following endoplasmic reticulum, and then in the cytosol. The way in the endoplasmic reticulum where they undergo modification and processing. Moving on to the next point, and till now over 100 gibberellins have been identified and then labeled as GA1, GA2 and GA3 and so on. And among these, GA3, that is called as gibberellic acid, that stands out as the most extensively researched plant growth regulator, and this is very important for the plant growth. Which one? That is GA3, that is gibberellic acid. Moving on to the next point, functionally gibberellins play a very diverse role in plant growth and development. So that's why these are plant growth promoters. Okay, plant growth promoters. So it is going to help in the plant growth and development. And what are the functions here? If I have to mention the functions here, these gibberellins can able to break the seed dormancy. What is seed dormancy? Seed dormancy means inactive state. The seed is going to stay in inactive state, but this gibberellin is going to break that seed dormancy. Means it is going to make that seed to germinate. It is going to make that seed to active. Okay, so that's why the main function of the gibberellins here is that it is going to break that seed dormancy. Promotes the cell elongation. Means if you think it is going to promote the cell to get enlarged or elongated, and it also promotes the cell division, and it influences the enzyme induction, and it also promotes the flowering and fruiting process. So these are the main functions of the gibberellins. Now we will move on to the definition. The definition is like this. Gibberellin is a class of diterpenoid plant hormone that regulates various developmental processes including the stem elongation, germination, means seed germination, flowering and dormancy. That is the simple definition of the gibberellin. We are moving on to the occurrence here, where we can see these gibberellins. They can concentrate in the regions like shoot apex, young leaves, embryos, and in the flower buds, and in the fruits and immature seeds. In all these regions, we can see the gibberellins. That what it shows, it means that it is going to found in the rapidly growing and developing regions of the plant. In these regions, we can find the highest concentration of the gibberellins. Moving on to the next point, they also have been found in algae, mosses, forms and gymnoforms. So in not only in plants we can find this gibberellin in algae, mosses, forms and gymnosperms. We will move on to the most important one, that is biosynthesis. Biosynthesis or gibberellins are synthesized inside the plastids of immature seeds and in young leaves and even in the roots. There you can all in all these places the gibberellin biosynthesis is going to take place. Thus gibberellin biosynthesis starts with the isoprenoid unit, that is called as IPP, and this is very important here. In exam they might ask this question like which is the precursor of gibberellin biosynthesis. Isoprenoid unit, okay, that is IPP. IPP means isopentyl pyrophosphate, or you can say isopentenyl diphosphate. Biosynthesis includes mainly 3 stages, with it is going to happen in a 3 different cellular compartments. Okay, in the first stage it is going to happen in a cellular compartment, that is in a plastids, and in second stage in the endoplasmic reticulum, and in the third stage it is going to happen in the cytosol. Gibberellin, firstly it is going to start in the plastids. I have labeled here. From here to here it is going to take place in the plastids. It starts with isopentenyl pyrophosphate, which is a 5-carbon molecule. This IPP undergoes a isomerization to form DMAPP. What is this DMAPP? The full form of the DMAPP is dimethyl allyl diphosphate, which is also a 5-carbon. So after isomerization of the DMAPP, the one more isopentyl pyrophosphate is going to add. Then this DMAPP is going to convert into a geranyl diphosphate, which is a 10-carbon molecule. GPP, it is converted, DMAPP is converted into a GPP, and then next, in the next step, again one more isopentyl pyrophosphate is going to add to the, and then it is converted into farnesyl diphosphate, which is a 15-carbon. And again for this FPP, one more isopentenyl pyrophosphate is going to add, so that it is going to convert into a very important one, that is geranyl diphosphate, which is a 20-carbon molecule. So you have to remember this molecule. This molecule is also very important. In the exam they will ask geranyl diphosphate. This GPP is converted into a copyl diphosphate by an enzyme called as copyl diphosphate synthetase, and then later this copyl diphosphate is converted into a end-corin by an enzyme called as end-corin synthase. All this process is going to take place in plastid. It is going to start in plastid. So you have to remember that is the first stage. Now we will move on to the second stage, that is endoplasmic reticulum, by an enzyme called as end-corin oxidase, which is simply called as KO. And this end-corinol is also converted into end-corinol AL by an enzyme called as end-corin oxidase. And later this end-corinol is converted into an end-corinoic acid by a KO enzyme, and this end-corinoic acid is converted into a 7-alpha hydroxycorinoic acid by an enzyme called as end-corinoic acid oxidase, that is called as KAO. And then later this 7-alpha hydroxycorinoic acid is converted into a GA12, that is gibberellin 12 aldehyde, and this gibberellin 12 aldehyde is converted into gibberellin 12 by an enzyme KAO, that is end-corinoic acid oxidase. All these steps are going to take place in endoplasmic reticulum. And also see here, I will tell one tip to remember this very easily. It's like, flow chart ide, so messy agide, nange ನೆನಪಿಟ್ಕೊಳ್ಳೋಕೆ ಕಷ್ಟ ಆಗುತ್ತೆ. ಆ ಥರ ಏನೂ ಇಲ್ಲ. ಸೊ ಅದಕ್ಕೆ ನಿಮಗೆ ಇಲ್ಲಿ ನೋಡಿ KO ನೀವು ಈ ಚಾರ್ಟ್ಸ್ಗಳನ್ನೆಲ್ಲ ನೆನ್ಪಿಟ್ಕೋಬೇಕು ಅಂತಂದ್ರೆ ಒಂದು ಕೀ ಪಾಯಿಂಟ್ ಏನು ಅಂದ್ರೆ, ಫಸ್ಟ್ ನೀವು ಫೈಂಡ್ ಔಟ್ ಮಾಡಿ ವಿಚ್ ಇಸ್ ಸಿಮಿಲರ್, ಏನೆಲ್ಲ ಎಂಜೈಮ್ಸ್ ಸಿಮಿಲರ್ ಇದೆ ಅಂತಂದೇಳಿ ಫಸ್ಟ್ ನೋಡ್ಕೊಳ್ಳಿ. ಅಂಡ್ ದೆನ್ ಸೊ ಆ ಸಿಮಿಲರ್ ಇರೋದೆಲ್ಲ ನೆನಪಿರುತ್ತೆ ಅಲ್ವಾ, ಸೊ ಅವಾಗ ನೀವು ಯು ಕ್ಯಾನ್ ಲರ್ನ್ ದ ಡಿಫ್ರೆನ್ಸ್. ಓಕೆ, ಡಿಫ್ರೆಂಟ್ ಆಗಿರ್ತಕ್ಕಂಥದ್ದನ್ನ ಕಲಿಬಹುದು. ಇಲ್ಲಿ ನೋಡಿ 3 ಅಲ್ಲಿ KO ರಿಪೀಟ್ ಆಗಿದೆ. ಈ ಆಕ್ಸಿಡೈಸ್ ಕಾರೆನ್ ಇಂದ ಕಾರೆನಾಲ್ ಇಂದ ಕಾರೆನೈಲ್ ಕನ್ವರ್ಟ್ ಮಾಡ್ತಾ ಇದೆ. ಈ ತರ ಸಿಮಿಲಾರಿಟೀಸ್ ನ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ ಫಸ್ಟ್, ಅಂಡ್ ದೆನ್ ಡಿಫ್ರೆನ್ಸ್ ನೀವು ಈಸಿಯಾಗಿ ನೆನ್ಪಿಟ್ಕೋಬಹುದು. ಇದು ಒಂದು ಟಿಪ್ ಐ ವುಡ್ ಲೈಕ್ ಟು ಮೆನ್ಷನ್ ಹಿಯರ್ ಟು ಮೇಕ್ ಯು easy for you guys. Now moving on to the third stage, that is which is going to take place in cytosol. So cytosol alli you can see here this GA12 is converted into a GA53 by an oxidase GA13 oxidase enzyme, and then this GA53 is converted into GA44 by an enzyme called as GA20 oxidase enzyme, and then this GA44 is converted into GA19 by an enzyme that is GA20 oxidase. See, illela similar agide. GA20 oxidase, GA20 oxidase, GA20 oxidase. And then this GA19 is converted into GA20. And then this GA20 is converted into a GA1. So which is very important. It is the one, GA1 is the growth, plant growth ge help agodu. Okay, GA20 is converted into GA1 by an enzyme that is GA3 oxidase. So ee minor difference na ನೀವು ನೆನಪಿಟ್ಕೋಬೇಕಾಗುತ್ತೆ. And then GA20 is converted into GA29 by a GA2 oxidase. Okay, not 20 oxidase, GA2 oxidase. See, these are all important because see, while exam ali questions yav tara indirect ಆಗಿ ಕೇಳ್ತಾರೆ ಅಂದ್ರೆ mutations, like mutations, if there is a mutation in GA2 oxidase, what happens? Questions will be like this. GA2 oxidase mutant ಆದ್ರೆ ಏನಾಗುತ್ತೆ, GA20 oxidase mutant ಆದ್ರೆ ಏನಾಗುತ್ತೆ, ಪ್ಲಾಂಟ್ ಲಾಂಗ್ ಆಗಿ ಗ್ರೋ ಆಗುತ್ತೆ, ಪ್ಲಾಂಟ್ ಏನಾಗುತ್ತೆ ಲಾಂಗ್ ಆಗಿ ಗ್ರೋ ಆಗುತ್ತೆ. ಯಾಕೆ? ಬಿಕಾಸ್ GA20 ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ಬೆಳಿತಾನೆ ಇರುತ್ತೆ, ಸೊ ದಟ್ ಅದು ಲಾಂಗ್ ಆಗಿ ಬೆಳೆಯಕ್ಕೆ ಶುರು ಆಗುತ್ತೆ. ಇಫ್ ದೇರ್ ಇಸ್ ಮ್ಯುಟೇಷನ್ಸ್ ಇನ್ ದ GA2 oxidase, okay, you can say here the GA2 oxidase. And GA2 oxidase, here also it is there. So GA2 oxidase is going to convert GA1 into GA8. Okay, GA1 ಕೆಲಸ ಏನು, ಗ್ರೋತ್ ನ ಪ್ರಮೋಟ್ ಮಾಡುತ್ತೆ. ಸೊ GA1 ಹಾಗೆ ಇದ್ರೆ, GA8 ಆಗದೆ ಇಲ್ಲದೆ ತರ ಕನ್ವರ್ಟ್ ಆಗದೆ ಇರೋ ತರ ಹಾಗೆ ಇದ್ರೆ ಏನಾಗುತ್ತೆ, ಸೊ ಪ್ಲಾಂಟ್ ಗ್ರೋ ಆಗ್ತಾನೆ ಇರುತ್ತೆ. So ಈ ತರ mutations ಮೇಲೆ questions ಬರುತ್ತೆ, you should be smart enough to answer. Okay, so this is one side. And on the another side, GA12 is converted into GA15 by an enzyme that is GA20 oxidase, and it is converted into a GA15 is converted into GA24, and GA24 is converted into GA9. Okay, and this GA9 is converted into GA4 by an GA oxidase 3 oxidase here. Okay, 3 ಇದು ಡಿಫ್ರೆನ್ಸ್ ಇದೆ ಇಲ್ಲಿ. 3 oxidase, GA9 ಇಂದ GA4 ಗೆ ಕನ್ವರ್ಟ್ ಮಾಡೋದು GA3 oxidase. And then this GA4 is converted into GA1. So ನಮಗೆ ಫೈನಲ್ GA1 ಬೇಕಲ್ವಾ, which is a growth promoter. So GA4 is converted into GA1 by a P45 oxidase. Okay, this is the phytochrome 453. Okay, so and also here GA4 is converted into GA7 by phytochrome 451, and GA7 is converted into GA3 by a phytochrome 453. That's about the Gibberellin introduction, definition, occurrence and biosynthesis. In the next video, we will learn about the Gibberellins functions, and I'm hoping you have enjoyed this video, and thanks for watching.